Good morning, everyone. Myself, I'm Dr. Pawar, presenting today's the embryology lecture. Unfortunately, we reached up to this stage due to the present the COVID situation. No problem. Anyhow, we'll continue since ahead until the situation becomes clear with this online lecture series. So, as you heard the lecture, like previous lectures in which we have covered the fertilization, then we have seen the formation of germ layers. So, we'll continue the further changes in that. Already all this part we have seen. We are not revising all that again. We have seen all the steps in fertilization. We have seen results of the fertilization. We have seen what is the test in baby. We have seen how the sex is determined. After that, this further development, we have seen cleavages. So what is morula? That we have seen last time. Then we have seen the blastocyst. Then we have seen the blastocele, embryonic pole, embryonic pole. Then zona pellucida, we have seen what is its role. Then we have seen the formation of three germ layers, the endoderm, ectoderm, and mesoderm. Then further development, we have seen how they are forming. Then amniotic cavity, amniotic fluid, amniogenic cells, all that we have seen. In primary yolk sac, secondary yolk sac, extraembryonic mesoderm, along with coelom, we have seen. We have explained last time this diagram with various parts. Then we have seen amnion and what do you mean by chorion, that also we have seen last time. Then coming to the next part of the development, further development, we will see from here the formation of procaudal plate. Now, what do you mean by procaudal plate? This is a new terminology here. At once, the circular area near the margin of disc, the cuboidal cells from the endoderm, which is from lower side, it becomes columnar. And once it becomes columnar, it is referred as procaudal plate. It is a head end. Once it is converted into the procaudal plate, it is considered later on as a head end. It divides the embryonic disc into the two equal halves by forming the central axis. So this procaudal plate formation, it is nothing but the change occurring toward the endoderm cells. Initially they are flat, but they become cubical, and later cuboidal to columnar. So this changed area we call as procaudal plate, and that is future head. In this diagram, you can see diagram A and B. In the A diagram, this is surface view, embryonic disc seen in surface view. You are seeing from the top. Here, if you take a section at XY plane, you can see here the ectoderm, and this is endoderm. Endoderm, which is initially flat, then it becomes cubical in nature, and ectoderm it is in the form of column. What we defined previously, the procaudal plate. So it is nothing but here changes taking place in the cubical endoderm. You can see here they becomes columnar, and this changed area we call as a procaudal plate. So once this procaudal plate is formed, right, so it is head end. So endodermal cells here become columnar, and this circular area, so it is referred in future as a head end, and exactly opposite to it, it is a tail end. So central axis get formed. Central axis is now apparent. If you take a section at this level, you can see here the procaudal plate. The changes takes place in endodermal epithelium. This endodermal germline. And this is ectoderm. So this part it becomes from cubical to the columnar cells, and we call as procaudal plate. Formation of primitive streak. Soon after the formation of procaudal plate, the change occurs in endoderm. Some cells near the tail end of the epiblast proliferate and forms elevation called as primitive streak. Now we have seen change which occurs in the endoderm we call as procaudal plate. Now similarly toward the tail end, that changes occurs in ectoderm, and that is referred as primitive streak. Then formation of intraembryonic mesoderm. The proliferating cells from the primitive streak pass sideways, pushing themselves between the epiblast and hypoblast. We know that epiblast is ectoderm, hypoblast is an endoderm. So this intraembryonic mesoderm, which get formed by proliferation from the primitive streak, and primitive streak is nothing but the change taking place in the ectoderm towards the tail end. So from there cells multiply and passes sideways and pushing them in between ectoderm and endoderm. That is the intraembryonic mesoderm. These cells form intraembryonic mesoderm. As it is the second time form, so it is called as secondary mesoderm. Primary extraembryonic mesoderm we have seen in the previous lecture. So here you can refer as secondary mesoderm also, or you can say simply intraembryonic mesoderm because it is going within the embryonic disc. The same explanation we can see with the diagram. So this is procaudal plate, already we know it. Now this the primitive streak. So ectodermal cells proliferate here. So you can the sagittal section you can see here this XY plane. So this ectodermal cells toward the tail end, it proliferate and the multiply cells, number of cells, excessive cells here after proliferation from columnar cells. This area we call as primitive streak. And from here the intraembryonic mesoderm get formed. So this is the main source for the intraembryonic mesoderm. You can see here in this diagram, the next like A diagram here. So this is the primitive streak. After multiplication, changes take place from ectodermal cells toward the tail end. They pushes sideways and it goes in between ectoderm and endoderm and it is going sideways. So in this sagittal section, you can see here, this is a primitive streak. Some ectodermal cells get modified. They form proliferation and form a nodular structure we call as primitive streak. So rest of part of ectoderm here you can see, rest of part of endoderm you can see, this is a tail end. And from here, the proliferating cells goes in between these two germ layers, ectoderm outside, endoderm inside, and in between that it is running. So that is forming mesoderm. And it is called as intraembryonic mesoderm or secondary mesoderm. Here, the sagittal section taking in along with the extraembryonic mesoderm, this developing embryonic disc here. Again, you can see here, this is extraembryonic mesoderm, this is extraembryonic coelom. Due to that coelom, it is split into two parts, somatopleuric and splanchnopleuric we have seen. Already in last lecture, we have seen amniotic cavity, embryonic cells and all that. This is ectoderm. Now the change occurs here in the primitive streak. So some cells of the ectoderm toward the tail end, they proliferate and form primitive streak. And that is the main source to form intraembryonic mesoderm. This is endoderm. This is the yolk sac, right? Endoderm multiplies here. And toward the head end, right, it forms a procaudal plate. So these are the changes, further changes in the developing embryonic disc. You can see here, this extraembryonic coelom, it expands. And this way, initially this portion is here, but it shifts toward the side by, and that is forming a connecting stalk. In connecting stalk also, right, this intraembryonic mesoderm splits, it goes here also. So you can see a labeling connecting stalk. Note that intraembryonic mesoderm passing into it. So this is a connecting stalk, and this is a future umbilical cord. So it gets elongated, it changes, shifts its position, and it forms long elongated structure, tubular structure, and that is called as umbilical cord. So connecting stalk is a future umbilical cord. So till now, we have seen a lot of changes in the developing embryonic disc. So ectoderm, endoderm, and mesoderm, three germ layers are formed. When primitive streak and procaudal plate it is formed, when connecting stalk is formed here. So it is a future umbilical cord. Now there are certain names to the developing embryo at this phase. One is called as embryon gastrulation phase. So when we are supposed to call the gastrulation, the process of formation of primitive streak. When the primitive streak get formed, endoderm get formed and intraembryonic mesoderm get formed. So at this phase, we can say the embryo is in the phase of gastrulation. The buccopharyngeal membrane, another term to understand here. Now we have seen a procaudal plate, which is modification right in the endoderm toward the head end. So at procaudal plate, the intraembryonic mesoderm does not go. So intraembryonic mesoderm from proliferating cells of the primitive streak reaches here, but it does not pass at the procaudal plate. It goes side of it. So only ectoderm and endoderm in contact with each other at this place, and that forms one membrane which is called as buccopharyngeal membrane. So this membrane, where the ectoderm and endoderm are in direct touch with each other, remains as it is. So this is a junction where the buccal means oral cavity, pharynx is part of GIT, and this buccopharyngeal membrane later on get ruptured, and then oral cavity is connected with the esophageal and rest of part of the GI tract. In that way, this buccopharyngeal membrane, by that time it protects the anterior end of the or opening of the primitive gut or GI tract. So this is buccopharyngeal membrane. Simply at this membrane, it remains membrane-like structure. So mesoderm does not enter at this area. So ectoderm and endoderm remains as it is in touch with each other. Cloacal membrane same way at the posterior side or tail end. This change occurs. Similarly, at tail end, intraembryonic mesoderm does not goes in circular area. There only ectoderm and endoderm remains in touch with each other. So mesoderm doesn't enter in between that. Same way like the head end. Toward the tail end, we got the cloacal membrane. So this membrane also get ruptured, right? Once the posterior end of the GI tract form, rectum and anal canal, that time this cloacal membrane get ruptured and the stool comes out. So in this way, both ends of GI tract are protected, right? Anterior by the buccopharyngeal membrane, posterior by the cloacal membrane. So in this diagram, you can see this is a procaudal plate anteriorly toward the head end. You can see here, this is a cloacal membrane. So anteriorly the procaudal plate, posteriorly cloacal membrane. You can see here, this is a primitive streak. So cells get proliferated from the modified ectoderm cells and intraembryonic mesoderm get formed. This intraembryonic mesoderm, it goes sideways, reaches here, but it doesn't enter in this procaudal plate. Similarly, toward the tail end, this does not enter into this area circular, which is called as cloacal membrane. So simply, we can say at the both ends, right, anterior and posterior end of the primitive gut later on, so this mesoderm is not entering here, and they remains as a membrane where ectoderm and endoderm, they are in touch with each other. Similarly, here you can see another change, intraembryonic mesoderm, that also pushes into the connecting stalk. So extra mesoderm is connecting stalk, in that intraembryonic mesoderm also enters, and this is the site, right, where the umbilical cord get formed later on, right, in the further stages of development of embryo, or we can say the fetus. So if you consider this diagram, we should know that what is the procaudal plate. We should know that primitive strip later on get elongated, right, and the posteriorly we get cloacal membrane. Now here another one structure we come across is a developing notochord. So this primitive streak, so cells get proliferate. Initially it is nodular, circular, then it becomes elongated, right? What we call primitive streak, and then some cells, right? So below that, right, get modified or get stimulation, and they will form, right, the further notochord. Later on, the primitive streak gradually elongates in anterior posterior direction along the central axis of embryonic disc and become a pear shape. Already in last diagram you have seen, it becomes a pear shape diagram, primitive streak. When embryonic disc is formed, first it is suspended in between amniotic cavity from above and yolk sac from the below or down, and connected to the trophoblast outside by the structure which is called as connecting stalk. So connecting stalk, why we call connecting? Because it connects a developing embryo to the trophoblast, which is outermost covering there. This connecting stalk, in future, it gets elongated, it becomes tubule-like structure, and this is nothing but the umbilical cord, which is the main connection between the placenta and developing baby. So by that time, the placenta get developed, right? And then it has a connection with the developing fetus with the umbilical cord. Umbilical cord has a lot of functions. So it provides nutrition, it supplies blood supply to the developing fetus. Same way, the excretion waste products were collected or transported through the umbilical cord to the placenta. And then through the mother circulation, it is excreted out. So umbilical cord plays very important role. At the time of parturition or the delivery, we have to cut this umbilical cord, and then it becomes a umbilicus in adult person. Initially, it is broad. Later on, as embryo elongates, it becomes small in size and shift to the tail end. So one can understand this connecting stalk. There is a shifting. Initially, it is in the central part, then it shift to the one side, then it goes to the tail end of the embryo. So regarding this connecting stalk, umbilical cord, again we will talk right in the future lectures, right, a lot on that. Then coming to the cloacal membrane, already we have seen that. What do you mean by cloacal membrane? One thing is clear, it is toward the tail end. So it is the posteriormost end, we can say, of the primitive gut. Toward the tail end, intraembryonic mesoderm passes toward the connecting stalk. That time it leaves small circular area where it does not passes. So ectoderm and endoderm, similar like the buccopharyngeal membrane. So at the procaudal plate, here also it intraembryonic mesoderm does not pass below the ectoderm or in between the ectoderm and endoderm. So both remains in touch with each other, and that is called as cloacal membrane. This is toward the tail end. So that is called as cloacal membrane. At the end, when the delivery takes place, by that time, all the parts of the GI tract get formed. So this cloacal membrane gets ruptured. Then and then anal canal is open to the outside. At the same time, buccopharyngeal membrane also, before that, it gets ruptured. So the esophagus part of the anteromost part right, of the GI tract, it opens or it has a connection, communication with the oral cavity. So in this way, the buccopharyngeal membrane and cloacal membrane protects the embryo, protect the developing fetus early phase. And once the delivery occurs, they get ruptured and they connect this GI tract or tube-like structure anterior to the mouth and posterior to the outside through the anal canal. Coming to the stem cells, one should know that what mean by stem cells, because stem cells therapy that is coming nowadays. So it is most popular toward the western side. It is very costly treatment nowadays, right? It is we are hearing these names right, little bit in India. So one should know as a part of embryology what mean by stem cells. The inner cell mass is multipotent. If you go to the retrospectively, the first phase after fertilization implantation, right? So trophoblast get multiply, right? And the inner cell, once the trophoblast get modified, the remaining part we call as inner cell mass. And these cells, right, inner cell mass, after the so many cleavages, right? So they are multipotent in nature. They are pluripotent. They have a lot of capacity to convert into the what are the cells that required for the development of various tissues. So in that way, it is called as multipotent or pluripotent. It can convert into whatever the cells required in body. So that's why it is called as multipotent. It can convert into any cells we want or the body need. We can store it under the proper condition for the longer period if we provide a proper environment right, to that cells. So such stem cells that we can preserve it for longer period. And if we are able to take precaution of the stem cells for longer time, so we can replace the stem cells whenever we want to replace that disease part. So stem cell therapy, and this is called as stem cell therapy. So stem cell therapy is a new concept in which, so this multipotent early part of the inner cell mass, right, we can store it for longer time. And they can utilize to replace what are the disease part cells. So this is called as stem cell therapy. So one should know that what do the stem cells and what is the stem cell therapy in short. Coming to the another terminologies in the further stages of development of embryo, one is called as pre-organogenesis period. So period from fertilization to the formation of bilaminar disc. So when the ectoderm and endoderm, so it is in the form of two layers, that is called as bilaminar embryo. So that period, that mostly first 14 days, it is called as pre-organogenesis. Organogenesis means by that time organs get formed. So, but the period before the formation of organs, we call as pre-organogenesis. If congenital anomaly occurs, if during this time something goes bad, something goes wrong, that time it is decided that death of embryo takes place. So embryo will not survive right, for the further development. So there is a death of embryo will take place, cells will get die. And in this way, pregnancy will not continue. So this is the most sensitive period, pre-organogenesis period. If something goes wrong, maybe in the form of trauma, germ, or any genetic problem, or exposure to the radiation or medicine, that time, right, as it is a delicate phase, so that time it will not be accepted by the developing embryo and the cells get died, and pregnancy may terminate at this early phase only. So we have to take a lot of precautions in the pre-organogenesis period. Then the gastrulation, already we have seen formation of the primitive strip, intramural mesoderm and endoderm. So that phase is called as gastrulation. Embryo is passing through the gastrulation phase, means one should understand these cells are get formed within the developing embryo. That is called as gastrulation. Then embryonic period. So from the 3rd week to the 8th week, that period is called as embryonic period. Most congenital anomalies occurs in this period. So from 3rd to the 8th week, embryonic period is referred, and most of the congenital anomalies occurs in this period. Now, one can notify, right, approximately, right, in development in days and what events occurs in the developing embryo. So, at the age of 2 days, age in days. So, by the age of 2 days, embryo is of 2-cell stage. By the age of 3 days, embryo, right, modular stage is occupied or achieved. That means 16-cell stage we call as modular stage. Then by the 4 days age, blastocyst formation takes place. By 8 days, the bilaminar disc, these 2 germlets, they get formed and embryos in the form of disc cell. By the 14 days, the procaudal plate and primitive strip get formed. Then by 16 days period, the embryo, it is intraembryonic mesoderm form and 3 layers of embryo form. So 3 germ layers. Right? So it is trilaminar embryo by the age of 16. So if you go in development in the days, right, you can see these changes which are occurring in the developing embryo or fetus. Now, apart from this, right, what we have seen till now, the further changes, there are certain another terminologies one should know that. So we will see that abortion. So with this new CBME course, right, the competency-based medical education, these things are added for this new syllabus. So one should know about the abortion. Once you know the fertilization, implantation, cleavages, then blastocyst, germ layer formation. So by that time, right, one should understood the terminology abortion. Abortion is the ending of pregnancy by removal or expulsion of an embryo or fetus before it can survive outside the uterus. That is before the 28th completed week of gestation. This is called as abortion. So it is the ending of pregnancy by removal or expulsion of embryo or fetus before it can survive outside the uterus. That is mostly like around 28 completed weeks of gestation before it. An abortion that occurs without intervention is known as miscarriage. So very early phase, if the pregnancy doesn't continue, we call as miscarriage. This miscarriage word is utilized, or spontaneous abortion. Within understanding, right, within a short time, that pregnancy gets terminated, and it is called as spontaneous abortion. When deliberate steps are taken to end the pregnancy, it is called as induced abortion. Maybe with the medical background, right, we can decide that before the actual ideal time, right, for the termination of pregnancy, you can plan it, and this is called as induced abortion. A similar procedure after the fetus has potential to survive outside the womb is known as late termination of pregnancy. This is a very late stage. If delivery occurs by the time the fetus is able to survive with some supporting system outside the uterus, by that time also, under the medical reason, we can terminate pregnancy, but it is more risky factor towards the mother regarding this termination of the pregnancy. And this is referred as late termination of pregnancy. Usually they are risky. So what we are seeing, abortion is termination of pregnancy. It may be of spontaneous, means sudden in early phase, or induced or planned abortion. What is the reason behind this? Etiology. Etiology means what are the causes behind it? In 50% of cases, there are genetic reasons. So abnormal karyotype. Karyotype means the chromosome analysis is done and their abnormal chromosomal numbers or abnormal chromosomes are there. So 50% reasons are there, 50% causes of MTP, medical term of pregnancy, they are genetic. Also, apart from that, remaining in 50%, we can say maternal factors. So mother may get infected by the torch infection, or endocrine factors, such as endocrine diseases, endocrine hormonal disturbance, immunological factors, maternal systemic diseases, anatomic defects, and trauma. It can also is also responsible, right, for the medical termination of pregnancy. Then toxic factors, certain the medicines, certain radiations, right, which are acting as toxic factors, they can also induce this medical termination of pregnancy or abortion. Pathologies, what are the diseases behind it? For certain pathologic conditions like hemorrhage, hemorrhage means bleeding into the deciduum basalis. So just below the placenta, right? So you get hemorrhagic changes, hemorrhage, bleeding, excessive bleeding due to the placenta get detached, and the nutritional part is get hampered, and that baby get died. And this is that embryos died, and that is the reason right, behind the medical term of pregnancy. Necrosis and inflammation, certain inflammatory conditions can also cause this medical termination of pregnancy. Uterine contractions and cervical dilatations, expulsion of most of all of the products of the conception can take place in this, and due to that abortion takes place. Coming to the last part of this lecture, one should know about the pregnancy test. And this test is depending on one of the hormone which is secreted in early phase when embryo it is of 5 to 10 days. So once the period is missed, so in next 10 days, this HCG human gonadotrophic hormone it slowly increases. It is secreted by syncytotrophoblast, and this can be excreted out through the urine. So what is this pregnancy that we can detect, and that can determine that one can come to know, right? So the pregnancy is there or not. So this is referred as pregnancy test. How to do it? Take the test of 5 to 10 days once you miss your period. Means by this 5 to 10 days, if you immediately on the next day if you do it, right, you will not get confident, or we can say the positive or proper results. So by 5 to 10 days after missing of your period, you can do this pregnancy test. Now how it is done? In this, the urine sample is collected. So collect the morning sample is most reliable because overnight sleeping, right, and hormones they are secreted properly. So in morning urine samples, hormones can come properly. So collect the morning urine sample, but you just let it right to pass the first part of urine, and then in the middle you can collect that part we call it midstream urine, and that should be collected in clean container so that contamination will not be there and it can not deviate the result. Add the urine sample to the marked pit or absorbent tip using a dropper. So you have to put a drop of urine, which is a midstream urine collection. Leave the pregnancy test undisturbed and check the result after stated time. So everything is mentioned on that pregnancy test kit. You should wait to take a pregnancy test until the week after you miss period for the most accurate result. If you want more accurate results, wait for 5 to 10 days after the missing of period. If you are pregnant, your body needs time to develop detectable levels of HCG. Yes, they are formed initially, but they are not at detectable levels. So HCG by 5 to 10 days, it is coming to the quantity, such quantity which can be detected. If if the pregnancy test is positive, the test can usually detect this hormone in your urine about 10 days after your first missed period. This is when the fertilized egg attaches to the uterine wall. During the first 8 to 10 weeks of pregnancy, human gonadotrophic levels normally increases very rapidly. So once it starts forming, initially it is very slow, but later on it level increases rapidly. Human chorionic gonadotrophic hormone or HCG is a placental hormone initially secreted by cells. The names of the cells is syncytotrophoblast. Now, in the next lectures, we are going to see the development of placenta. By that time, we will come to know what we mean by syncytotrophoblast. So these cells get formed by the time, and they secrete this HCG hormone, human chorionic gonadotropin hormone. And by the 10 days, it is detectable in the urine sample. So by cells from the implanting conceptors during second week, supporting the ovarian corpus luteum, so and which in turn support the endometrial lining and therefore maintain the pregnancy. So this hormone level is useful to maintain the to continuous pregnancy, and these cells they are supporting, or this hormone is supporting in continuation of corpus luteum to maintain that pregnancy. Coming to the conclusion of this today's lecture. So here we finishes the pregnancy test. here is a summary, right, of the lecture, right? From the first fertilization we have seen, results we have seen, goals of fertilization we have seen, blastocyst, then abortion and all that in between, right? Spontaneous and the induced abortions, then pregnancy test. All this can be, right, for the pregnancy test, what is the anatomical principle behind it? So all this may ask, right, for the 5 marks short notes, right, in brief on the following. So in this, you are supposed to define it. In this, you are supposed to explain it properly with the help of diagrams. So this can be asked the questions that concern with this topic. So here we finishes this extended lecture in continuation with that fertilization. So what are the further changes? Mostly in the second week, right, second or third week, what changes are occurring in the developing embryo that we have seen. Thank you. Next time we'll mostly, right, we'll see, right, how the development of placenta takes place. Once again, you read this topic from the book. What are the difficulties, right? You can convey through the message, right, if you don't understand. But my request will be that, right, you read from the different books or whatever the books you are following, right, and see the diagrams, take the help of diagrams, you will understand properly. Thank you.